And thank you all for coming. And please feel free to stop me and ask questions if points that I'm making are unclear. I have had about 3 different graduate lectures from my own course, Botany 682, that I've cobbled pieces together. So I don't want to lose anybody as I jump from one topic to another. What I'd like to do today are to have a discussion about the biology of these reef plants, because I think there is a growing interest in using them as indicators for the health of the reef. And I want to make sure that you folks at least are able to understand the complexity. Even though the plants are apparently simple in their form, the complexity that they have is not necessarily simple. I want to run through some of the prevailing models for reef dynamics so that you can start to see how ecologists think about reefs. I give a brief summary of some of the older work from Maui, where we've been studying algal blooms since about 2002, and then point to at least some future endeavors that are, I think, some of the projects that are already ongoing, or have projects that other students have done that I think have some real traction from my perspective as a marine botanist interested in the physiological ecology of seas. So that's, I hope, where we go. I have about 160 slides, so we may not get through the whole lecture.
Well, we have the 5. So
Yeah, that's what I figured. Now,
keep these guys alive.
a lot of the newspaper coverage that deals with algae would make you think that whenever you have an abundance of a seaweed on a beach like this picture, it's a devastating impact, it's a non-natural situation. In fact, this is a picture from the late 19th century, so that's the 1800s, with a prominent landmark in the background and piles of limu that people are actually picking seaweeds out of this pile of limu ele, which is an alga species, and limu lipoa, I think, is the other one that they find in this area. So having piles of algae on the beach doesn't necessarily mean that we've had dramatic impact in a coastal environment, at least in a way that is unnatural over the last 100 or so years. Many of you may be familiar with the limu from the kind of cultural perspective. If you're actually from here, you may know about the Hawaiian cultural practice of ho'oponopono. Is that familiar to anybody? Right? Okay, this is a symbolic interaction among family members when there's strife within the family. The Hawaiian cultural practice would be to go to the beach and collect some of this plant, limu kala, bring it home and put it on the dining room table, and that symbolizes the need for the family to reconcile these issues. Kala meaning to forgive. Right? So the cultural knowledge that we have about seaweeds is actually quite well developed and documented through my colleague Dr. Isabella Abbott's activities. She wrote about these plants for the last 20 years of her life. We also know that there are plants like limu vaiole. This is another favorite food alga that would be something that you could find in Tamashiros or some of the markets that are down in the Chinatown area for sale that you would chop up with your tomatoes. So some of you, if you've had poke and limu poke, you've already started to interact with these plants in the ocean. Right? There is a kind of cultural basis that we have with them. But do you know what their life history is like? Right? In the simplest form, those last 2 genera that I illustrated have the same life history that we have, in that the dominant plant, this limu kala, is a diploid entity. It has 2 sets of chromosomes, just like we do. The gametes are produced via meiosis. Reproduction takes place externally to the plant in the water column. This very big egg is fertilized by a very small male gamete that swims to the egg. There are perfumes that the egg puts out in the water to attract the male to the egg. It's a very sophisticated means of reproduction, as you would expect, right? It's the same system that we have. This is called gametic meiosis. That very big egg now, in botany terms as a zygote, it settles within 6 cell divisions, is already established a rhizoid for attachment, and the photosynthetic progenitor cells that give rise to the whole rest of this upright plant. This will grow in a year. That's the typical kind of biomass that you see for limu kala. This is a genus that in Waikiki in the 70s was about 1 meter in height. This is a very successful plant that grew in conditions where the nutrient levels were background ambient nutrients. There weren't excessive amounts, and yet you have big biomass fields of this particular genus representing a very healthy reef. So the first message to disabuse you of is that if you see lots of algae, it clearly doesn't mean that an area is sick. It does mean that there may be native communities that are highly productive and adapted by evolutionary pressures to be successful in those regions. And newspaper coverage kind of forgets that part. It's like to focus on the algae that cause all the problems, like in Kaneohe Bay or in Waikiki now. So that's one life history, but in Botany 480, I can torture students endlessly with the life history. Here's genus Gracilaria. We have some very common species, Monowea here, and the long ogo here. These are edibles. They are sell for, with ahi, or $15 or $16 a pound at Costco. Right? So this is your ahi poke. We're looking at the algal partner to that. The life history for this plant is very different than Codium or Sargassum. And if you're using it as an experimental design, you need to know that the plant you're collecting in the field could be either this diploid plant with 2 sets of chromosomes like the Sargassum had, or these haploid plants with only 1 set of chromosomes. And they look identical. You cannot tell them apart. You have them in your hand. You cannot tell them apart. And yet, because we have so little knowledge about them, there's every reason to think that there may be differences in the way they respond to the environment, the way they respond to submarine groundwater discharge, based on whether it's the sporophyte, the botany term for these 2 sets of chromosomes per cell, or the gametophyte, the plants over there that produce the gametes. So, this is called an isomorphic alternation because the phases look equal in their morphology, iso meaning equal. Questions on that? You cannot tell them apart in the field. You go down to a Kaalabai and make some collections. That is the story then for Gracilaria.
It's still the same species though.
Same species, absolutely. And this is where it's genetically identical, except for this difference in the phase that they have that is obligate for them to go through a complete sexual cycle. What many of our invasive species do, this is the same genus as the gorilla ogle that dominates parts of Waikiki, is that many of these will just forego the sexual reproduction and branches will break off of either this phase or this phase and attach themselves because they're very simple, doesn't take long, a few days, maybe they're putting out anchoring rhizoids, and what was a branch a week ago is now a new plant that's starting to grow. And so we have a kind of clonal propagation that becomes a mechanism for them to become so dominant. Like at Wailupe, if you've seen the submarine, the boil that's out there, Gracilaria salicornia is in a large dense mat around that boil. That is probably a clonal, not going through this life cycle, but a kind of iterative process with these plants breaking off. Water motion is enough to break them. That's basically all they need. But if you don't know that, that's a problem in terms of your experimental design. Right? What does it mean to sample if all your plants are genetically identical? How are they replicates? Oops. I think these are issues that I can bring to you as students interested in submarine groundwater discharge to try and better the quality of the work that's being done.
Well, how are you going to deal with it?
What we've done is actually run genetic assessments. So you take a transect from the boil and you go out at certain distances and you sample periodically, and hopefully you can detect a difference across that gradient. Or you at least explicitly sample over larger distances so that the chances of having a different genotype, different genetic individual show up are enhanced by the scale of the sampling rather than assuming that each plant that you pick up is genetically distinct, because it's likely it's not. So these are maybe fine scale issues, but I think it points to some of the adaptive strategies through evolution that these plants have evolved that allow them to be so wildly successful when supplied with sufficient nutrients through a boil like we see out at Wailupe for the groundwater discharge. You've got one adaptive strategy that's very well suited to life in that setting, and the water motion breaks them up and they just spread and become the dominant species out there, the dominant even genotype out there. Pretty cool. Okay. Gracilaria is one, but this life history is actually common to most of the brown algae, like Diopterus or Limolipoa, or green alga like ulva, which is another one of our reedy species that you find in places like Algae, Black Point area, where there's a little spring that's right below Doris Duke's home. And so all of these plants have that kind of isomorphic alternation of generations. You have gametophytes and sporophytes co-occurring on the same bench. You can't tell them apart. For this one, at least, you have the capacity to tell them apart when you bring them back into the lab and examine them under a microscope. You can see the swimming cells that they are producing about every 3 days. These things are so fecund, they're producing probably 1 million swimming cells per centimeter squared of tissue. That's a lot of spores or a lot of gametes that go through the life cycle. This is one of the reasons why this plant is so productive and so commonly found, is that it floods the market with its reproductive units. It's got those two kinds, the spores and the gametes, that are both competing for space on the ring. Okay, any questions on that? Okay, so this kind of alternation of generations is common. It is the life alternation, or the life history that algae have. And if you're going to use them as a tool, as an indicator for reef processes, you really need to pay attention to the issues that are tied to the biology of these organisms. One of the plants that throws a wrench even in that complexity is this plant, one of the most prized of the Hawaiian seaweeds, Limokopu. This is the plant part that's collected by many practitioners as they're going across the islands. There's only one name for kopu as you go across all the islands. It's very common and well known. It's been collected by pinching off these uprights, and then you can find this at Haile's on Kapapulu, down again in Tamashiro's little rounded balls of those upright axes that have been cleaned and salted and set on the side to aerate. They sell them for like $5 or $7 for a little ball of that stuff that's about this big. It's a very potent flavoring agent. People love it to mix with their poi and other foods. There's a funny story about an organic chemist who looked at the chemistry of the compounds that Hawaiians were eating in this way and couldn't believe that they actually were okay after eating it because of the known toxicity associated with those compounds. So they'd done the chemistry, they extracted it, identified it, published it. He looked at it and said, there's just no way that they could eat this and be healthy. So he came out here, went to the beach, found some, ripped off some upright axes, popped them in his mouth, felt so ill. A few minutes later, they rushed him to the hospital. So how is it that Hawaiians could eat this stuff and not suffer that same effect? Because of course they don't get rushed to the hospital every time they eat limu. It's the most common, one of the most common foods that's left in their limu diet. Any ideas? That aeration process that's sitting on a table for a couple of days in the shop and after cleaning is enough for the volatile components to evaporate so that the levels that are left are spicy but not dangerous. This individual couldn't believe that he got sick from eating this stuff, so his next trip out, guess what he did? Same thing over again. Found the plant, picked up some axes, chewed on it, boom, back in the hospital. So part of why I wanted to start this lecture with the kind of cultural context is that there is a lot of traditional ecological knowledge in the Hawaiian culture about the distribution of various limu and where you commonly find them and the practices that are associated with their growth and the timing, and a lot of information that obviously this individual didn't respect. Right? And so he learned the hard way that some of this information probably has some real merit associated with it. What else is so interesting about Asparagopsis taxiformis is that its life history stage alternates with this little pink fuzz. Right? This is the diploid plant, the sporophyte, the plant that has double chromosome count per cell. These are the gametophytes. Right? And so we now have entered into some of the more complex bases where if you were, as an ecologist or somebody interested in submarine groundwater discharge, wanting to talk about the impact of a discharge on Asparagopsis, you would, and only sample this, you would only be sampling half of the actual diversity that is contained within this alga. You would not be doing a full job, you would be doing only a part of it as a basis for the study. So where do you find these little pink fuzzes, this Falkenbergia stage? This is a microscopic view. They're little tiny pink fuzzes, and the places that these are commonly found are in reef regions as epiphytes on other annual plants like Limucala, the sargassum that could grow to this size. Right? So they are in the ecosystem, but at a different level than the macroscopic plant that has some differentiation in terms of their ecology. You might expect that their photosynthetic rates, their growth rates, maybe their response to submarine groundwater discharge are actually quite different because their form is so different. The ulvas and the Dictyopteras and the Gracilarias might be expected to be more similar because their forms are so similar. They find comparable ecological areas. So this gives you a little world, a little bit of insight into some of the complexity when you start going out to sample from our flora and hoping that it's a good tape recorder for what's actually going on out in the reef. This is the, excuse me, the gametophyte phase on this side. It produces reproductive units that are diploid spores that fall out of the plant and settle to the bottom. This is the little Falkenbergia pink tuft that via meiosis produces these upright axes that grows into the lumicola. Okay, so biology is really a fundamental component in a well-designed study. You have to know what you're doing. It's not just that you can run out to the beach and pick up Gracilaria salicornia and think that it's going to be all okay. While I've got your attention on this, I want to just take a minute and point out some of the real gems in our flora. These are plants that have been collected only a few times. Like this Dudresnia babitiana was collected up at Midway one time at a 20-meter depth. It's a plant that's big enough for a herbarium specimen sheet. This is about 20 centimeters up here in the scale. This is presumed heteromorphic and is also an endemic. So in our flora, some of the plants that you may come across are plants that are not found outside of Hawaii. Some of the plants that you find as you were to go to Waikiki are going to be very commonly distributed outside of Hawaii as well as here. So this is one of our jewels. Here's Dudresnia littleri, another plant that's an endemic. Halaminia, another plant that's endemic. It's not accidental that many of these are all red algae. We have a number of them that are in that category of being endemics. So our flora, for you to use as tools, is rich. We have over 550 species of native algae. But that also poses real challenges for you as experimentalists. You might want to try to get in and study what these algae can tell you about the health of the reef. Some of the earliest data that we have comes actually from Jeff Polovina's studies looking at the energy flow out of a reef, and this is probably one of the best reasons why studies of algae are so important in understanding ecosystem dynamics at a broad level. The benthic algae at French frigate shoals, through Polovina's assessment, actually were the predominant biomass producers. They are the plants of the ocean. They feed the heterotrophic benthos as well as all of these herbivores, either directly or indirectly. So essentially, because of the order of magnitude greater productivity because of the algae, all of this life exists. The phytoplankton, as you might guess from the clarity of the water in the northwestern Hawaiian Islands, this water is so clear, the phytoplankton don't hang around very long, they're exported with water motion. It's the plant production that's actually sustaining literally all of this by an order of magnitude greater benthic productivity than phytoplankton. So that's another reason why we'd want to have algae included in studies that talk about indicators for tropical reefs. I fuss over having all the benthic algae placed in one box. But what does one box tell you when you have 550 species? It's a bin in which you lose a tremendous amount of the diversity that I started talking about with the life histories. Or in other dimensions, say the palatability of some of these reef fish for eating these algae. And so early studies by one of my grad students, Jen Smith, will actually just simply ask the question, if an alga is on a reef, which of the fish in our reefs are going to eat them? Do all fish eat all algae, or do some reef fish eat only certain algae? That herbivory is one of the two regulating factors for the occurrence of plants on a reef. So here is a suite of reef fish that were selected for their studies. And you can see the acanthurids, those with little tiny mouth parts like the tanes have, are in the dark blue. The scarids, or the parrotfish with these big crunching jaws, are in the light blue. Do all fish eat all algae? These are the algae down here on the bottom. Clearly not. Clearly, some of these plants are heavily grazed and others are not eaten at all at very low levels. And so if you go to a site like Algae or Guadalupe and you only see one species, is that because the grazing pressures have eaten everything else and that plant is out here? And in fact, Gracilaria salicornia, the dominant plant at Guadalupe last summer, is right here. It's in that very non-preferred category. Maybe it has nothing to do with the nutrient supply. Maybe it's actually the herbivory pressure eating everything else. So you would want to know more than just the SGD, sorry Craig, but you want to know more than just the delivery of nutrients. You have to also think about the biological interactions that are taking place if cover on a reef is part of what you are asking in your experiments. So about at the same time Jeff Polovino was working, my colleagues the Littlers put together a paradigm or a hypothesis that tries to resolve this grazing pressure activity and nutrient supply, not counting for salinity, just nutrients, as a means to understand what this does to influence the plants that are in a benthic community. And so we could have the normal arrangement on a coral reef, a healthy reef with lots of coral. This is the Hawaii Visitors Bureau view, right, with lots of coral and lots of fish. Would be to have high grazing and low nutrients. What's the low nutrient level around here? Joan, do you have a number for us for low nutrients?
For
Waikiki.
open ocean seawater?
Yeah, ocean seawater.
Probably about 0.2 micromolar nitrate.
And maybe a 10th micromolar phosphate, something like that. Okay. So that, I would argue, is not a level of nutrient supply that would allow for a large algae to be successful to grow rapidly. These big plants have lots of surface area and they have lots of place to store nutrients in the cell volumes that are present in there. So maybe you wouldn't expect to see a lot of dominance in a region where there's a low nutrient delivery on a regular basis. But anything that were to show itself above the coral head would be eaten because the grazing pressure is high. So you have to now think about having all those reef fish that you see in pictures, hundreds of reef fish, they're in the way, you can't even see long distances in the pictures. Most of their stomachs are full of algae. Their grazing intensity is so significant that it might be as many as 2,000 bites per meter square per day. That's a lot of grazing, right? That's on par with trying to feed my 17-year-old when he's playing baseball, right? Pizzas disappear, ribeye steaks, porterhouse steaks disappear. This kid can eat, as these can when the number of fish is high. So, then we see, in conditions where there might be a reduction in the number of fish grazing, what we tend to see are algae that are illustrated here microscopically, and these are all microscopic pictures. They may be a centimeter in height. We call these turf algae. These are not going to be good for you to manipulate because they're very fragile plants. They are the kind that the tang eat readily. They are tiny in stature, but we have over 200 species in this. This is a very diverse community of plants that are certainly relying on the nutrient supply, but being cropped readily. You see them show up on a reef when the fish intensity of grazing drops off. How could that happen on a reef? How could you have a reduction in grazing pressure?
Fishing pressure.
Fishing, yeah, okay. Anything else? There could be seasonal, there could be movements of fish, there could be an increase in predators so that there's a natural drawdown in the number of fish. There could be failures in recruitment classes of juveniles coming onto a reef, so there could be natural processes, and then it could be human-induced processes like overfishing with support. But these things are so fast in their growing, in part because they're so simple, they can complete their entire life cycle in 2 weeks. Fast. This is like having the lawn in your front yard go, and then baseball season comes and suddenly your grass is now a foot tall and your neighbors are wondering what the heck happened to you. These are perennial players on the reef. You just don't see them because in a healthy reef with lots of grazing, they are the food for those fish. That's one group. The next group are these rock-loving or rock-like algae. These are getting good press, unlike the invasive algae. These living pavements, you can see them as these pink forms, can either be branched or they can be rock-like solid calcium carbonate structures with a veneer of living tissue over them. They're a lot like corals in that they have calcium carbonate in their cell wall, and in fact, if Linnaeus and his crowd in the 1700s originally thought they were coral, they placed them in with the animals, and then they realized about 100 years ago, there's no polyp. They don't have feeding parts. In fact, they're pink. They're not the usual brown that a coral is, and somebody had the bright idea, oh, they must be red algae. And so this order of corallins, coral-like algae, is actually really important on a reef because they are grazing resistant. Few fish, except the parrotfish, can chomp on them. Their mouth parts are strong enough to actually gouge that tissue. When the tissue is there, the coral larvae in their own life history actually seek those surfaces out. They have a chemical signal in the corallins that allow for corals to settle. So reef restoration often pays a lot of attention to the amount of corallin algae that are present because that's a suitable surface for the next generation of coral recruitment. Okay, so that's our second group of algae. We have the turfs, we have these coral-like ones, and then the ones that we use for submarine groundwater discharge work are those frondose algae, the big ones, the plants that can reach a meter in stature. We have over 200 species in this category of green, brown, and red algae. 70% of our flora is red algae. So these plants, because of their stature, have the cell volume to actually probably store a fair amount of nutrient. Pulses could come through the environment and they can actually acquire that and store that and use it for growth later on, independent of the nutrients in the water column. And I think that's actually a really important strategy that we realized and adopted in early submarine groundwater discharge work because we knew that if we were going to use them, we had to get the plants and then hold them in a nitrogen-poor environment in order for them to be forced to acquire what was in the plume around them at the time of the experiment start. Does that make sense? Kind of? Not really? If we just pulled them out of the reef because of their storage capacity associated with their large size, we were unable to get them to respond. They were living off their bank account. So we had to force them to spend their bank account, and then they would draw in the nutrients that were in the surrounding waters. Okay, so if we start putting all of this together, you can see that there are a couple of states, 4 alternate stable states on a reef, actually. Not just a coral-dominated reef, but 4 different states. Coral being one of them, but frondose algae as a dominant portion of the ecosystem is another stable state. We see that in northwestern Hawaiian Islands. There are vast fields of algae that are native in the most undisturbed community left on the planet for a coral reef ecosystem. These are really important issues because it gives us some insight into the role that nutrient levels play and why algae would be good indicators for change on a reef. We did those early experiments in a manipulative way Jen Smith and Peter Broom actually put out PVC tiles out on the reef. We added fertilizer by putting them in an unglazed ceramic pot. Put fertilizer in there and put that pot out, and it diffused into the environment. Here's the fertilizer with a little funnel. We looked at water motion through a cloud card, and out on the reef you can start to see the cages that would keep the herbivores out. And so in a simulated way, we actually tested these relative dominance ideas with an experimental design. That's published in 2001 in Coral Reefs. Here's the nutrient uptake work underway without any cages, so there's no grazing pressure present. And here's the nutrient plus the caging representing that final combination of low grazing, because there's a cage here, and elevated nutrients because of the flower pot. And what we found as a result of this in a short period of time, so I'm saying like within 6 months of the start of these experiments, going from PVC tiles like here, where the grazing pressure is keeping them clear, we found plants that, that look like this on the tiles. The analysis of this community led us to realize that within a short period of time, the reef community in a coral-dominated area like Puako, the reef community was responding with 4 different states, completely different states associated with the combination of grazing and nutrient supply. We could drive Puako's community into one of 4 different stable states. That, to me, says these reefs are not these vast, resilient ecosystems. Within 6 months of adding fertilizer, we can push them into a macroalgal-dominated ecosystem, as seen by what's on these little tiles. Okay. And I think that's an important perspective, that these ecosystem components become really quite responsive when the nutrient levels are changed by, say, a submarine groundwater discharge event or having other factors like wastewater treatment plants, on-site sewage disposal systems having plumes that come out of the coastal environments. When you couple that with a lowering in fish activity, you have the potential and what should be a coral reef to end up with something that's very different. Here's some variation. This one set of pictures isn't going to tell you everything, but a couple of sets of pictures show you that in some cases we got green algae and corallins, and some of the finer branched species, articulated corallins. Here are some of the corallins. Here are some of the other macroalgae. The community showed a lot of characteristics of being a responsive ecosystem with lots of different species involved, but always those same functional forms being repeated. This to me was really a revolution. I used to think, well, corals take 100 years or several hundred years to grow, but we're driving change for the settlers in a short period of time, in just a matter of months. This study went on in a paper in 2010 to actually track with what we call successional trajectories, to look at how those paths change as you go through time. This is just one snapshot in time, but we sampled every month. So we have 14 months worth of data to show that, in fact, it's not just one time, but it's throughout the development under these conditions that we see really dramatic differences in the communities that result after these factors have been set in place. Okay? So this is one reason why algae are good indicators on the reef. And if you have a submarine groundwater discharge plume, you're adding elements to those reefs that elevate the background nitrogen composition. The plant community, you should expect, will respond. Any questions? Okay. So let's put some of this together. I have this little, very simple cartoon, don't laugh. But I'm trying to illustrate what I see happens as you have open space on a reef and competition among the 550 native ocean plants, the 50 corals, and then other invertebrates that settle on the substrate like sponges and tunicates and other kinds of organisms. These spores or zygotes from the algal life history rain down. This is the prevailing paradigm. They rain through the water column and find a spot to settle. And this represents our typical diversity. We have about 60 or 70% red algae, and the other 10 or 20% are green and brown algae. So the first battle is just to get there. and find a spot that you can settle. The next battle is to have them start to grow. So symbolically, we're going to see that very quickly they would start to compete for light. But in part because these are different color groups, the functionality of their photosynthetic apparatus actually limits that kind of competition to only the red algae really competing for light among themselves. The green algae would not that well compete with the browns. They absorb different colors of light. This is where photosynthesis becomes a really important factor. But for nutrients, there should be ample competition, right? These are absolutely essential components for growth of these plants. It's not that they are able to eliminate or find a different part of the nitrogen cycle that they can take best advantage of. They all need nitrogen, and especially in large amounts because of the photosynthetic apparatus. So we might expect that if you have a sufficient level of nutrients that you would see rapid growth by all of the algae that are present based on their functional form. But under conditions of limiting nutrients, they would all be suppressed in their size, the growth rate, even the photosynthetic capacity. So this gives us different metrics that we can start to understand and measure in the field. Under those conditions of low nutrient, like we have in our typical coastal healthy reef settings, I would expect that those plants that have a high affinity for a particular flavor of nitrogen, like nitrate, those plants that have a high affinity, or in other words, a very efficient uptake system, should flourish. And I think this is one of those handles that we have that can give us a handle on the physiological traits that allow us to understand why certain species win. If one species is better at grabbing nitrate than another, that one that has that high affinity capacity is going to win in the pairwise comparison or pairwise competition. And so we could also have other species that have a very low affinity. They don't do well. They can't acquire the nutrients in order for them to grow at high growth rates. So our oligotrophic ecosystem, this low nutrient environment, really pushes some of these plants, I think, to edges of their competitive ability. This Ks value that I've illustrated here is actually biochemistry. It's looking at the Michaelis-Menten coefficient for enzymology, trying to understand the rapid rate and then the saturation for that uptake rate. So it's just half of the maximal velocity. So a low value means the plant's really responsive. A high value means it takes a lot. I think for future research, this is going to actually be an interesting parameter to pursue, trying to gauge in advance of a bloom what are the competitors that could win, because you have a nutrient pulse and a very efficient system, or a nutrient pulse and a not so efficient species. So those species with a high affinity would grow well. Those species with a low affinity wouldn't grow well under low nutrient regimes, which is the kind of pristine setting. How this interplays with submarine groundwater discharge, I think, is really exciting interface for future research. We also have herbivory, of course, and we have to consider that whenever we're looking at field conditions. We often forget about it, but it is absolutely regulatory in terms of its ability to modify species and abundance through the selective grazing or the generalist grazing that occurs in the leaf region. And so if you had grazing taking place that obviously is going to crop back the amount of a plant that's present, if it's selective grazing, so as I showed you, say the Gracilaria salicorneus, not well eaten by many of our reef grazers, over time what that's going to allow is, say, this non-palatable alga to increase in its fecundity, increase in its abundance, and over time you would see it become the dominant because it's not being grazed. So these are factors that have taken us really 20 years as biologists and ecologists to tease apart so that we gain some appreciation. So the favored foods are eaten, the non-palatable ones grow. If we look back at some of the old data from Waikiki, my colleague Max Doty, my predecessor in botany, had a bunch of grad students that periodically would go out and actually survey all the algae in this grid in front of the natatorium There are funny stories about a little Filipino grad student who was sent out to the outermost boundary with a stadium rod so they could estimate tide height, and he would get buried by the waves as they broke. And lots of funny stories about this. But if you take a look at the data, which he never did, and they just put it in a notebook. This is 1969. They had so much information they didn't know how to handle it. They looked, when I cracked those books and looked at them, the green algae all end up having a very distinct distribution pattern, as if the selective grazing or nutrient supply was not so sufficient out in these regions, but allowed for their abundance to reach high levels in this inshore region close to the distance from the shore. So this is 0, that's about 250 meters up there. So the green algae dominated in this region. If any of you were snorkeling, you're probably all too young at this point, but I used to snorkel in this area in the 70s, and it was alga reticulata everywhere. It was a green alga that dominated and snarled and everything. And so is this because of nutrients? Is this because of herbivory? We don't know. We don't have that background information. But it's interesting when you look at the brown algae, and this is largely the sargassum, like a little color that gets to be a meter in height. Its abundance was out here on the other end of the reef, where the water motion is greatest, although it was presumed to be at low nutrient level. So the plants, this native genus was able presumably to get enough nutrient to allow it to attain this large biomass over this portion of high water motion energy, high energy region of the reef, and be excluded likely from this region where green algae occur.
They're more robust.
They are mechanically stronger, yes. And they have a perenniating base that sends upright. So even if the upright got torn off by the water motion, they would send another one up. Whereas the greens are these sheer little blades that rip, and they don't have that kind of mechanical force or strength. So there are lots of reasons. But there could also be the flux of nutrients in this allowed them to sustain large biomass out here, and quieter water didn't give them enough nutrient, or it was already stripped by the time it got to these regions. So we have lots of interesting hypotheses and some historical data. Just a quick summary of that. We see that species matter. The sargassum are most abundant, these two taxa in the outer reef region. Green alga like Halimeda discoidea is most common in the inshore to the nearshore area. There are some that are present at low levels, like rare, but across all of the reef. And then the first documentation of the invasive Acanthophora coming in and being relatively high in abundance right up against the wall of the Natatorium. So this is an invasive red alga, one of the first to actually be documented in its arrival here. So that's what Limocala used to look like, a real forest. It used to be, you'd have to part the plants in order to swim through the reef waters in that region. It was like swimming through a car wash, right, with all those brushes that are coming down. That's what you get if you have this kind of density of biomass. That's what we should have. And now the invasive Gracilaria salicornia has dominated that entire reef, at least until the hurricanes started coming through, and now there seems to be much less biomass. Okay, I have a couple of other stories I want to tell, and one of them, before I get to the Delta 15N work that we've done, so Craig, stop me if I'm starting to get too long. I'm at about an hour mark here. How you have,
take a break at some point if people want to.
if you want, sure. Has anybody heard of the Redfield ratio?
Oh,
Uh-oh. My take on the Redfield ratio
pay
is,
attention.
Yeah.
This is like, elementary math.
well, we might have some differences of opinion, Craig. Then I think it really comes
I
from
didn't say it was I'm saying it's a fundamental concept.
fundamental. But it was, in fact, a unifying contribution for biological oceanography. What this gentleman, Redfield, did was get samples of all kinds of marine life from the open ocean. And the ratio of the atoms that are found in these organisms, here's the ocean conditions. You can see the phosphate versus the nitrogen, and these follow a very specific ratio. And that in the end, the atoms found, the atomic ratio found in all life in the open ocean followed this ratio, almost invariably. 106 carbon to 16 nitrogen to 1 phosphate. This has been held as a basis in marine plant communities to try and understand nitrogen limitation or phosphate limitation in those algae in coastal environments where the supply of nitrogen and the supply of phosphate through mechanisms like submarine groundwater discharge and others is much more dynamic than you would expect to see in the middle of any of these ocean basins. And so I found it really intriguing. As a plant physiological ecologist, I'm always interested in the adaptive strategies that the algae have to life in their environments. I would expect when nitrogen is available, these macro plants will acquire it, they'll stick it into photosynthetic pigments, they'll do all kinds of things with it. And so it would make sense to me that as you come close to that interface with land, where nutrient supply is readily available, that we would see significant divergence from this ratio. That makes sense? At least as a hypothesis. If we go and take a, here are some of the ways in which those molecules are used. The red algae especially have very high nitrogen requirements. They're pigments that make them red take 4 times the amount of nitrogen that a chlorophyll molecule takes in order for it to be functional. The nitrogen demands are enormous, and so you might expect for them to be very responsive to nitrogen supply in their ecosystem. Okay, so with work summarized across the published literature, we can start to see the 106 to 16 to 1, the regular ratio for these plants is not well represented or faithfully repeated. Here's one that's a little bit close. Here's another one that's fairly close. These top are cold water algae. Our warm water algae, where nitrogen availability in the background level is low, we would expect to see maybe increases in the amount of carbon for the amount of nitrogen that's present, and that's exactly what you see. A lot of photosynthetic carbon in response to, in comparison to the amount of nitrogen to phosphate. And these values are substantially distinct. We have some interesting adaptive strategies that are underway in looking at the red filtration. That is repeated in the brown algae, where the cold water and the warm water algae are still distinct, although maybe not quite so strikingly. Their photosynthetic apparatus doesn't use a lot of nitrogen. It has carotenoid-based molecules, as well as chlorophyll. And then the green algae that we have, again, shows marked differences from the temperate to the tropical algae. So I interpret these data to give us some insight as to how these plants actually are suffering from or taking full advantage of the environmental delivery of nitrogen and running photosynthesis to generate lots more carbon than we see nitrogen to phosphorus. So that's one piece of information that you can file away. We have physiological measurements that look for how nutrients in the nitrogen family, nitrate or ammonium, are acquired by plants. If this is the outer cell wall, and here's the plasmalemma of an alga, the incorporation of these molecules is active transport. We have molecules that are involved in moving these compounds into the cell. So suddenly physiology becomes really important. Nitrate reductase is an enzyme that actually takes the nitrate that you might see in a delta 15N analysis. The way that it gets incorporated into that tissue is through the enzymology of this enzyme. For ammonium, it's a different enzyme, glutamine synthetase. We look at the enzymology, it's interesting to see that we've got actually a 2-step process, nitrate reductase and nitrate reductase. These are the enzymes. Maybe this is a little bit too much, but what I want to show you is that the process for acquisition of nitrate is actually an enzyme, nitrate reductase, that is linked so intimately with the next stage that if there isn't photosynthesis running, this nitrite reductase won't run, and then the nitrate reductase won't run, and the alga won't even take the nitrate up. Right? Light has to be on. Photosynthesis has to be running. Nitrate as it is assimilated by an alga, regardless of whether it's submarine groundwater discharge or some other plume that's coming into their environment, is associated with daylight activity, photosynthesis running. That's different than what we see for glutamine synthetase, which can run in the background. This may be more than we really want to talk about, but the work by Ivan Valiela here, it was whole study this area, Cape Cod, where the groundwater has particular signatures and the rivers have different loadings. He looked at different algae for the enzymology. And what you can see is that for those plants with larger nitrogen loading, you can start to see higher nitrate activity, nitrate reductase activity. The plants are responding to the availability of that substrate. The ones that, from all of these studies, the ones that bloom are the ones that actually have the capacity to acquire those limiting nutrients. So now, excuse me while I jump through these. Here's the glutamine synthetase, alkaline phosphatase. If you're interested, this is older work, but it's still the best that we have for documenting the response that a community-based ecosystem has based on species and the enzymology. So in summary, the land-based loading has saturated the glutamine synthetase and the alkaline phosphatase. We see the nitrate reductase activity, especially in this Cladophora, as being substantial. And ironically, Cladophora is one of our bloom algae here. It was a bloom species on Maui, driven by land-based sources of pollution back in the pineapple days on the west coast of Maui. There is a fair amount of nitrate reductase activity for those areas. This is why nitrogen is so essential, and photosynthesis is linked to that. From that, growth becomes evident. By looking at land plants, so this is a sunleaf, a plant that sits in high light environments, as many of our algae do. 75% of the nitrogen that is taken up by those plants is tied up with photosynthesis in some way. So if you're going to be a plant and living in a marine environment, the nitrogen availability becomes a real limitation. If you are living in a nitrogen poor environment, photosynthesis and from that growth is going to be substantially limited. Okay? So all of this leads to a set of experiments that Scott Larned did for his dissertation. This is back in when there was a zoology department, not biology. He actually set up, for the first time, working with our algae, experiments where he varied the nutrient concentrations over a 10 to 12 day growth period and set the lightest if they were about 4 meters thick. So there are 1 control and 3 treatments: nitrogen enriched, phosphorus enriched, and nitrogen and phosphorus. So the goal was to try and determine out of these what the response of the plants were, and he surveyed about a dozen of our species. This is the first unquestioned, full, robust test for the role of nitrogen or the role of phosphorus as a limiting factor that would make them plants that would acquire these new elements, these limiting elements from various land-based sources of pollution. So the results that you would expect to see based on water quality, if you had nitrogen limitation, what he did was raise it from under 1 to over 6 for dissolving organic nitrogen. For the phosphorus treatment, he went up by 2 micromole from 1/10 of a micromole, and then the combined treatment of NNP. If it was nitrogen enrichment, you would expect to see rapid growth in this treatment if it was, as well as the combined treatment. If it was a phosphorus enrichment, you would expect to see larger in this one as well as in the combined treatment. So that's pretty simple, right? Very standard kind of growth measurements done in aquaria in order to allow for control. Here are the data. How would you interpret this for Caulerpa racemosa, one of our green, common, sand-loving plants?
What are we looking at here?
This is growth rate under control, phosphorus added to 2 micromoles, so control at 1/10 of a micromole. So it's 2 micromoles. This is 6 micromoles nitrate with background phosphorus, and then the 2 combined. So elevated both N and P here.
Nitrogen's one that
Yeah,
has the
clearly,
biggest impact for growth.
clearly, nitrogen is limiting growth of this plant, right? Here's Caulerpa, a second species, so for two species in the same genus, same pattern, nitrogen is limiting the growth of the plant. These are really important data, and I offered this in part because there have been other oceanographers with whom I've had discussions about the role of phosphate limitation, limiting reef activities broadly. There's no evidence for phosphorus limitation here, right? We've added it, it doesn't respond. The plants are responding to the nitrogen that's in these ecosystems. Nitrogen elevated allows for increased growth rate. Okay, so this is one genus, two species. Let's look at some of the other taxa that are present. Here's Podina, the brown alga, and another brown alga, Sargassum. This is the big reef plant. So Podina, clearly nitrogen limitation, right? For Sargassum, some increase in phosphorus, but a marked increase with nitrogen and even a little bit higher, although not statistically distinct, when you add the two combined. So the conclusion for this is that there's clear nitrogen limitation for these reef plants as well, growing in just the ambient water from Kaneohe Bay, and then these elements added to them.
These are all tank experiments.
Correct.
Yeah.
Replicated through time, and controlled flow rate, controlled light and temperature would not have been controlled, it would have been ambient water. Gracilaria salicornia, my friend from Waikiki, that's in quotes, can actually grow at a moderate rate under controlled conditions. So this is potentially one reason why it does persist, as well as the fact that it's not readily eaten. Give it a little phosphorus, it doesn't distinguish from the control rates, but add nitrogen and boom, you see a doubling in that rate. And put the two together, there still is a substantial increase. Haprophytus, this is the big alga in Kaneohe Bay that's coral dominant, overgrows corals and smothers them. Nitrogen response. So again, nitrogen limitation. We have throughout all of these studies, only one alga that he showed that had a phosphorus limitation. That was one of the codiums. And the explanation for that, when he looked at those tissues, the algae actually had heterocystic blue-green algae growing on the surface of the codium. Why is that important? A heterocystic blue-green alga can fix its own nitrogen and release it to the surrounding environment. So those plants of codium didn't need any more nitrogen. It was being supplied by the epiphyte living in that plant body.
So is this symbiotic?
It's associate. Right? It's not intimate, it could be brushed off, but in an ecological setting, it's there, and nitrogen in the form of ammonium leaks out, and the plants acquire it, so they have their own little nitrogen factory built in.
Maybe this would be a good time.
This might be a good
Take
time.
5 minutes.
That sounds good. And
Grab
it's-
some more cookies. These restrooms.